You know, what if I told you that the single most important conversation in the entire universe is actually happening inside your head right now? Today, we're going to decode that conversation. We're talking about the secret language your brain cells are using to chat with each other every single second. So, for the longest time, this one question just completely stumped the greatest minds in neuroscience. Is the brain basically one giant continuous web of tissue? Like a massive interconnected superhighway? Or is it more like a chain of islands, like an archipelago, with trillions of tiny, separate islands that somehow have to figure out how to pass messages between each other? Okay, so back in the late 19th century, you had these two giants of neuroscience basically squaring off over this exact question. In one corner, you've got Camillo Golgi. He developed this incredible staining technique and came up with what's called the reticular theory. Basically, the idea that the brain is all one big continuous network. But then, in the other corner, you have Santiago Ramon y Cajal. And get this, he actually used an improved version of Golgi's own method to argue the complete opposite. He called it the neuron theory, that the brain is made of individual, separate cells, and the real trick was figuring out how information could possibly jump from one to the next. So, who was it, the superhighway or the islands? Well, let me tell you, the answer was going to change absolutely everything we thought we knew about how the brain works. Believe it or not, it took all the way until the 1950s with the invention of the electron microscope to finally get a clear answer. And what they saw, just like you're seeing here, proved that Ramon y Cajal was right all along. Neurons are individual cells. They're separated by these tiny microscopic gaps. Turns out, the brain really is made of islands. And that special meeting point, that's what we call the synapse. And the actual physical space between the neurons is called the synaptic cleft. Now, this discovery just blew the whole thing wide open with a brand new question. If the cells aren't even touching, how on earth does a message get from one to the other? And that brings us to the next great mystery in our little detective story here. We've got to find the messenger. What is it that can actually cross that gap? Yeah, let's go back to that island analogy for a second. Imagine you've got an electrical signal traveling down one neuron. That's our first island. How does it get across the water, the synaptic cleft, to the next island? I mean, it can't just swim, right? It can't just jump. It has to send some kind of messenger. So the first big clue, this came from the absolutely brilliant work of a scientist named Bernard Katz. He was studying the junction where nerves meet muscles, and he noticed something really fascinating. Even when the nerve was totally resting, not doing anything, his instruments were picking up these tiny little random electrical blips. It was almost like he was eavesdropping on the faintest whispers of a conversation. And he gave these whispers a name, miniature endplate potentials. And this discovery led Katz to this just revolutionary idea, the quantal hypothesis. He basically proposed that the chemical messengers, the neurotransmitters, they weren't just flowing freely. No, they were stored and released in these standardized little packets. And he realized that each one of those tiny whispers he detected, that was the sound of just one single packet being let go. So a full-blown nerve signal, that wasn't some continuous stream, it was the sudden, perfectly timed release of hundreds of these packets all at once. Okay, so how much messenger are we talking about in one of these packets? You ready for this? Each tiny little packet, which we call a vesicle, is stuffed with about 10,000 molecules of neurotransmitter. That is an incredibly efficient and really potent delivery system. And here's the best part. When the electron microscopes finally got powerful enough, scientists were able to zoom right in on the synapse. And what do you think they found? Yep, tiny little sacs filled with chemicals, all lined up right at the edge of the neuron. These were the vesicles, the messengers in a bottle that Katz had brilliantly predicted years earlier without ever having seen one. Just incredible. All right, so we know the message is stored in these little packets, and we know what happens when one of them gets released. But that just leads to the next big mystery, doesn't it? What gives the command? What tells hundreds of these things to release all at once in perfect sync? This was the next huge piece of the puzzle to solve. I mean, how do you get this perfectly synchronized, massive release of neurotransmitters at the exact moment the electrical signal shows up? What's the trigger? What's the go signal? The answer, it turns out, is one single, absolutely crucial ion, calcium. And it all happens in a flash. So first you've got this electrical signal, the action potential, that just zips down the neuron. When it gets to the end, to the terminal, it basically throws open these special gates just for calcium. Now, because there's a ton more calcium outside the cell than inside, it just floods in. And that sudden rush of calcium, that's it. That is the direct trigger that causes all those vesicles to fuse with the membrane and dump their chemical messengers into the gap. It really is that simple. No calcium, no message. You can have the electrical signal arrive, you can have all the vesicles packed and ready to go, but if that calcium doesn't rush in, the message stays locked inside. It's the essential key that unlocks the entire chemical signal. Okay, so we've gathered all the clues in our little neuroscience detective story. Now, let's put it all together. Let's watch the entire process unfold from beginning to end. So here's the full play-by-play. The electrical signal travels all the way to the end of the neuron. This forces those calcium channels to fly open. Calcium rushes in. That calcium influx triggers the vesicles to merge with the membrane and release all their neurotransmitters. Those little messengers float across the gap, they lock onto the receptors on the next neuron, and boom, a brand new electrical signal is created and the message keeps right on going. And that right there, that is the elegant genius of this whole system. It's this beautiful, seamless conversion of information. The brain takes a super fast electrical signal, translates it into a precise chemical message to cross that gap, and then, instantly, translates it right back into an electrical signal on the other side. This electrical to chemical dance is the fundamental language of your entire nervous system. Now, just stop and think about that for a second. This tiny, unbelievably fast process we've just walked through, it's happening trillions of times, every single second, inside your brain. It's the foundation for every thought you'll ever have, every memory you create, every action you take. So the final question really is, with all of that incredible conversation happening inside you right now, what are you going to do with it?